So the next important step that we want to talk about cancer cells is that cancer cells have the ability to undergo angiogenesis. Angiogenesis is the process by which cells can actually call out or send out chemical messengers that will promote the growth of new blood vessels. Angiogenesis is a really important part of our growth and development. It plays a very important role as we grow. It plays a very important role in fetal development. So as an embryo is growing and developing, we're going to need blood vessels to supply that individual. And the interesting thing is that cancer cells are able to send out chemical messengers that will stimulate angiogenesis. They can actually call out red blood vessels to grow towards them and feed them. And so this is one of the dangerous things about cancer cells. Not only do they sort of grow in place and push everything out of the way and just get larger and larger in size, but they can cut off supply lines and nutrients because they can actually call out new blood vessels and capillaries to grow towards them. Once they're able to really do this, these cancer cells are also going to be able to start to metastasize. Metastasis is when those cells are going to cross that basement membrane, and they can invade blood or lymphatic vessels, and they can form new tumors in foreign regions. This is why it's so important to stop cancers at an early stage, because what was once lung cancer can eventually appear in your liver, can appear in your bones, can appear in other parts of the body. If those cancer cells can break off from the original tumor and enter into the bloodstream, they can travel through the bloodstream, through the cardiovascular system or the lymphatic system to reach other areas. So, cancers are going to ultimately result from genetic mutations, and usually those genetic mutations involve checkpoints in the cell cycle. So like we've already mentioned, is that cyclin is going to help direct the movement of the cell through the cell cycle. It's going to be responsible for saying, okay, the cell is good to go, it's good to progress to the next stage. So we look for certain things at those checkpoints. We're going to be looking for things like DNA damage. We're going to be looking for, do we have all of the proteins necessary to replicate the cell? Do we have everything we need to progress? If we don't have those things, then the cell does not progress. The cell doesn't undergo mitosis or meiosis. It's not going to split and divide until either the damage has been repaired or the cell is going to undergo apoptosis. It's going to shut down, and it's not going to continue to grow. However, if we have mutations in those checkpoint proteins, if we have mutations in the individuals who are responsible, or the proteins that are responsible for those checkpoints, the cell is not going to be able to have any control over the cell cycle. That cell is going to start to undergo rapid cellular replication, and it's just going to keep reproducing and cycling through that cell cycle over and over again, which can lead to cancer. There are two classes of checkpoint proteins. There's proto-oncogenes. Proto-oncogenes code for proteins that promote the cell cycle. So those particular proteins are responsible for saying, okay, we've got the go-ahead, move forward. There are also tumor suppressor genes. Tumor suppressor genes are going to inhibit the cell cycle, and mutations in both of these can result in cancer. You could have proto-oncogenes permanently switched on, so the cell is just always going through. Proto-oncogenes, you can kind of think of those as stepping on the gas. And what you have in your car to help control your car and make sure that you stop and go and don't run a red light, you've got the gas and you've got the brake. Two separate pedals with two different points. The proto-oncogenes are the gas, and it is possible to get those permanently switched on. So it's kind of like putting a lead brick on the gas pedal, and it's just going to drive forward. Now, that's bad enough in and of itself, but if you still have your brake pedal, you might be okay. You might have some control, but the tumor suppressor genes are sort of like the brake pedal. If there's a mutation in the tumor suppressor genes, if they are just switched off, if they are defective or inactive in any way, there's no stopping the cell cycle. So it's very important that we don't have mutations in our proto-oncogenes or our tumor suppressor genes, because a defect in either one of those can lead to cell cycle mutations and cell cycle lack of control over the cell cycle. Just like if you only have a gas pedal, you can let up off the gas, you can push down on the gas, but if you don't have a brake, you're kind of screwed. You don't have a whole lot of control over stopping. Same with your tumor suppressor genes. You can put your foot on the brakes, or you can take your foot off the brakes, but if you don't have the gas pedal, you're not really going to get that cell going anywhere. So the tumor suppressor genes and the proto-oncogenes are both very important to the cell cycle. When those proto-oncogenes mutate, they can become cancer-causing genes called oncogenes. Even though we really only possess 2 copies of each proto-oncogene, only one needs to be mutated in order to lose control of the cell cycle. Some proto-oncogenes code for growth factor or their receptor proteins. So they do play a very important role in regulating that cell cycle. We do need them. But when those proto-oncogenes become oncogenes, the receptor proteins are easy to activate. So in other words, it's just like having a really sensitive gas pedal. It's really easy to get it turned on, very difficult to get it switched off. When those tumor suppressor genes mutate, their products no longer inhibit the cell cycle, or they don't have the ability to promote apoptosis. They can't shut it down. So even if they are able to detect that there's a mutation, even if they're able to detect DNA damage, they can't actually shut the cell off. So unlike proto-oncogenes, both copies of this gene have to be mutated in order to completely lose the cell cycle. So a mutation of the tumor suppressor gene BAX. So just an example is a mutation of the tumor suppressor gene BAX. Its product, the protein BAX, is going to promote apoptosis. If BAX mutates, apoptosis is much less likely to occur. The tumor suppressor protein, you may have heard of this one, P53, activates DNA repair enzymes, and it can turn on genes that stop the cell cycle. So it can promote repair of that DNA damage, and it can also switch on, or it can turn off the cell cycle. Unfortunately, what we see a lot in tumors is that a lot of tumors are lacking the P53 gene, or they're lacking P53 protein altogether. So there you go. There are many examples in your textbook of the genes that are specifically involved. I strongly would like to encourage you to read through that just to see if you're familiar with any of the genes that are involved in tumor suppression or involved in tumor growth, but we will not be quizzing over specific genes or their specific gene products. So we do want to mention just briefly in this overview different types of cancer. Oncology as a whole is the study of cancer. An oncologist is a medical specialist in cancer. The patient's prognosis is simply the probable outcome. So a diagnosis is sort of what disorder the individual has. A prognosis is what is the likely outcome of that disorder. So an individual's prognosis is going to depend on whether the tumor has invaded surrounding tissues and whether there's metastatic tumors in distant parts of the body. So like I said, when my mother was diagnosed with breast cancer, that was the very first thing they looked at, is what were the margins on that tumor? Was it nice and round? Had it started to invade? And then was there potential for cancer in other parts of the body? We did have a very close family friend when I was young who died of cancer. He had undergone several rounds of chemotherapy over the years. He had gone into remission probably 4 different times. And each time it just kept cropping up in other areas. So it started off as a lung cancer, but then they thought they got it all. They had removed all the tumors. He had undergone chemotherapy and radiation. And then the next thing you know, it had, like 2 years down the line, it had appeared in his liver. And so then they cut it out. They removed the tumor. He had undergone chemo and radiation. And 2 years down the line, again, they found it in his stomach, and it had just kind of kept spreading throughout the body. At that point, when there are metastatic tumors in distant parts of the body, at the point where it's now spreading throughout the rest of the body, it becomes much harder to control, much harder to stop. So, like I said, there are different types of cancers. That's just going to be classified based on the place of origin. So, where did they originate? So carcinomas are going to be cancers of the epithelial tissues. Sarcomas are going to be found in the muscle and connective tissues. Leukemias are cancers of the blood. Lymphomas are cancers of lymphoid tissue. Blastomas, those are cancers composed of immature cells. So really young, not fully formed cells. Lung cancer is going to be the most common respiratory system cancer. It's going to be one of the most common types of cancer as a whole. And typically this is going to arise from smoking. Our good family friend who did have lung cancer, he actually had never smoked a day in his life. So where it had come from, the original origin of that lung cancer was not known. But one of the most common causes of lung cancer is smoking. Smoking also can increase the risk of oral cavity cancer, cancers of the mouth. Colorectal cancer is another really common form of cancer. There are other cancers of the digestive system like the stomach, pancreas, esophagus cancers. In the cardiovascular system, some of the cancers include leukemia, plasma cell tumors. The lymphatic system, cancers are going to be classified as either Hodgkin or non-Hodgkin lymphomas. And that's just going to depend on whether they are coming, those lymphomas are coming out of mutated B cells or coming from B or T cells. Thyroid cancer is going to be the most common type of cancer found in the endocrine system. So really I have just a few more. The most common reproductive system cancers are going to be breast and prostate cancers, but there are also cancers of the cervix, ovaries, testes, and penis. The urinary system cancers include bladder and kidney cancers. Skin cancers include melanoma and basal and squamous cell carcinomas. So really, I'm not going to quiz you on those. I just want you to understand that cancers can be found in all of the systems, all the different systems of the body that we're going to talk about. Every single one of them is susceptible to cancers because every single one of those systems is made up out of cells. It's involved with cells and tissues and organs. And anywhere there's growing and dividing and replicating cells, we're at risk for cancers. So you should be able to list the characteristics of cancer cells that allow them to grow uncontrollably. You should be able to describe how mutations in the tumor suppressor genes and proto-oncogenes regulate the cell cycle. For your own purposes, you should be able to identify some of the more common types of cancer. Honestly, you can probably do this just based on what you know. But this is not necessarily going to be something that I'm going to quiz you over, so you don't need to memorize or worry about those, but just be familiar with those lists and where and how cancers can arise in the various systems of the body.